Hey guys, welcome to SpecTransfer and to topic 3.1.4.2, many proteins are enzymes from the AQA A-level Biology specification. As always, let's start off with a look at our specification. So we should know that enzymes lower the activation energy of the reactions they catalyze, as well as the induced-fit model of enzyme action. We should know that the properties of an enzyme relate to the tertiary structure of its active site and its ability to combine with complementary substrates to form an enzyme-substrate complex. We should know about the specificity of enzymes, as well as how the rate of an enzyme-controlled reaction is affected by enzyme concentration, substrate concentration, concentration of competitive and non-competitive inhibitors, pH, and temperature. So let's make a start. Enzymes are proteins that act as biological catalysts. This means that they speed up chemical reactions by providing an alternative reaction pathway of lower activation energy, but they are not used up in the reaction itself. Enzymes hereby reduce the activation energy required for the reaction, which increases the rate of reaction. So just to define activation energy is the energy needed for a reaction to start. In all reactions, bonds are broken in the reactants and new bonds are formed to give us the products. Energy needs to be put in to break bonds at the start of the reaction, and this is the activation energy. The lower the activation energy, the easier it is for a reaction to happen, and therefore the faster the rate of reaction. As you can see in this energy profile diagram, there is a lower activation energy in the reaction with the enzyme. Therefore, we have a faster rate of reaction. So how do enzymes lower the activation energy? This is explained by the lock and key model, which you have learnt at GCSE, and the induced-fit model, which is a development on the lock and key model, which is the additional model that we'll need to learn for A-level. So just to recap the lock and key model, which you'll have learnt for GCSE. The tertiary structure of the enzyme is such that the shape of the enzyme's active site is complementary to that of the substrate, so the substrate can fit into the enzyme's active site to form an enzyme-substrate complex. At A-level, we have to learn the induced-fit model, which proposes that in order to completely form an enzyme-substrate complex, the enzyme and substrate have to alter their shape slightly. This weakens specific bonds in the substrate needing to be broken, which lowers the activation energy required for the reaction to happen, increasing the rate of reaction. Next, we'll have a look at the effect of a few factors upon the rate of enzyme-controlled reactions. First of all, temperature. As the temperature increases, the rate of reaction increases. This is because enzyme and substrate molecules have more kinetic energy, so the frequency of collisions between these molecules increases, so more enzyme-substrate complexes are formed per unit time. However, after the optimum temperature, the rate begins to decrease. This is because the rise in temperature causes hydrogen and ionic bonds, which hold the enzyme's tertiary structure in place, to break, altering the tertiary structure of the enzyme, changing the shape of the active site too. After a certain temperature, the shape of the active site is no longer complementary to that of the substrate. The enzyme is said to be denatured. We also need to know about the effect of pH on enzyme-controlled reactions. This graph shows how rate of reaction changes with pH for a number of different enzymes. They all have different optimum pHs. Different enzymes have different optimum pHs. For example, salivary amylase has an optimum pH at pH 7 because it's secreted in the mouth, whereas pepsin, which is an enzyme that digests proteins, has an optimum pH at pH 2 because it works in acidic conditions in the stomach. As you move away from the optimum pH, rate of reaction begins to decrease. This is because the change in concentration of H+ and OH- ions interferes with hydrogen and ionic bonds that hold the enzyme's tertiary structure in place, altering the tertiary structure of the enzyme, altering the shape of the active site too. If the pH deviates too far from the optimum pH, the shape of the active site is changed so much that it is no longer complementary to that of the substrate. The enzyme is said to be denatured. Next we have enzyme concentration. At A, the more enzymes there are in a solution, the more likely a substrate molecule is to collide with one to form an enzyme-substrate complex. So at first, enzyme concentration is directly proportional to the rate of reaction. However, after a certain point, as we can see at B, the amount of substrate becomes limiting, so increasing the enzyme concentration has no further effect on increasing the rate of reaction, so the rate levels off and remains constant. Next we have substrate concentration, which shows the same trend as enzyme concentration. The more substrate there is in a solution, the more likely an enzyme is to collide with one and form an enzyme-substrate complex. Therefore, at A, substrate concentration is directly proportional to the rate of reaction. However, after a certain point, represented by B on the diagram, the amount of enzymes becomes limiting because all active sites are occupied, and therefore increasing the substrate concentration will have no further effect on increasing the rate of reaction, so the rate of reaction levels off and remains constant. Finally, we need to know about enzyme inhibition. There are two types of inhibitors that we need to know: competitive and non-competitive inhibitors. Competitive inhibitors are molecules that have a similar shape to that of the enzyme's normal substrate. They therefore can fit into the enzyme's active site to form an enzyme inhibitor complex. This prevents substrate molecules from binding to the active site. Competitive inhibitors compete with substrate molecules for available active sites. The difference in concentrations of the inhibitor and substrate molecules determines how many enzymes are inhibited. Note that the higher the concentration of competitive inhibitor, the lower the rate of reaction, because there is more competition for available active sites, so fewer enzyme-substrate complexes are formed per unit time. Next we have non-competitive inhibitors, which, unlike competitive inhibitors, work by binding to the enzyme in a place other than the active site. This changes the tertiary structure of the enzyme, altering the shape of the active site too, meaning that it is no longer complementary to that of the substrate. Substrate molecules can therefore no longer bind to the active site. Note that non-competitive inhibitors are often foreign to the body and toxic, such as cyanide. This is an irreversible reaction. The shape of the active site is forever changed. So what is the effect of a non-competitive inhibitor on the rate of reaction? Rate of reaction is decreased as non-competitive inhibitors reduce the number of functional enzymes available. Substrate and inhibitor do not compete for the active site, so increasing the substrate concentration has no influence on the effect of the inhibitor. When comparing the effects of competitive inhibitors versus non-competitive inhibitors versus no inhibitor at all on a graph of rate of reaction against substrate concentration, reactions in the presence of a competitive inhibitor are able to reach the same maximum rate of reaction as when no inhibitors are used, just it requires a higher substrate concentration to reach this point. This is because by increasing the substrate concentration, the competitive inhibitor can be outcompeted by substrate molecules, whilst non-competitive inhibitors, on the other hand, reduce the number of functional enzymes available. Substrate and inhibitor do not compete for the active site, so increasing the substrate concentration has no influence on the effect of the inhibitor. Great, that would be enzymes covered. We now know that enzymes lower the activation energy of the reactions they catalyze, and we've had a recap of the lock and key model and have covered the induced-fit model of enzyme action. We know that the properties of an enzyme relate to the tertiary structure of its active site and its ability to combine with complementary substrates to form an enzyme-substrate complex. We also know about the specificity of enzymes, as well as how the rate of an enzyme-controlled reaction is affected by enzyme concentration, substrate concentration, concentration of competitive and non-competitive inhibitors, pH, and temperature. That would be it for now, guys. Thanks for watching. Please subscribe, comment. Next time, we will be covering the structure of DNA and RNA.